using to amplify our genes. That's where we are mainly using the PCR primers to design our own. We need to design our own primers to amplify our chain of interest, right? Am I right?
Yes, ma'am. Yes, ma'am.
Yes. Then, okay, when we are having, it's not like everybody can, any sequence we can take and we can take that as a primer. We have some conditions for its primers and some temperatures we need to check. But like while doing in online, it is fine. Like you can get the results and you can face the results. While doing it practically, you will face like many problems like regenerating your like primers. You're not going to do your generated primers. You're just going to thank your primer generating companies. But the design you have to give, which gene part you want and which thing you don't want, those things you have to give, right? So for that one, we are having like it's a kind of an idea kind of thing. Like we'll have an overall idea of what is primer, how to generate that one in primer blast. Okay. Okay, let's move on to that one. This is from NCBI and this is called as primer designing tool. It's primer blast. So if you see here, this is the page of that one. Here you have to paste your sequence because you are going to create a template for your PCR product. And you see the primer parameters. Like from 5, if you are giving your own reverse primer or like those kind of things, you can give here. From 5 prime to 3 prime. While like developing a primer, we always have to design from 5 prime end to 3 prime end. Even if it is reverse primer also, we need to create a complementary pair for that one. Then only you can start from like for example, if I start coding from here region, it has to start coding from this region. So we cannot reverse from 3 prime to 5 prime, we cannot design. We need to design from 5 prime to 3 prime as a complementary sequence, then we have to match it. Okay, likewise you have to do. So consider this as your forward primer, this as your reverse. Between you are having your sequence. But forward primer, no need to complement it. Directly you can ATGC, those things you can type up to whatever amount you want. And here also in the reverse primer, while thinking of the reverse primer, you need to check. You have to complement it. Complement your like 3 prime into 5 prime. So so for we are having 1,000 base pairs we are having. So what about our primer length? How much it should be? Any kind of idea you are having about your primer length and all? Anybody has any idea like the primer length should be this much? It should not cross that much like that. Okay, then I will tell.
It will be 20 nuclear lengths, right?
Okay, yes, from 18 to 24 we can maximum we can make. More than that we cannot make your primer because it will pair up with each other. So that's why we are only going for a minimum number of nucleotide base pairs we are creating. So 20 is fine. At the below you can use 18. At the maximum you can use 24. Okay, that much of base pairs only it should have in your primer region. So for reverse also it is similar. You have to have that kind of from 18 to 24 reverse complementary like primers, and it should be of pairs. It should not be of like single digit codons. It should not be 19 because while generating like one primer will be left alone, so it will not bind with the like your original sequence. So it should be paired with each other. Like if you are having choosing 20, 20 base pairs, it's fine, and 18, it's fine. Don't go for 19 or 21 or 23. Those odd numbers, try to avoid that thing. Okay. And anyway, and the product PCR product size up to maximum of 1,000, which means the protein we are like amplifying region, right? Up to from this region to that region. If I'm using 1,000, like my protein, like my gene sequence is of 1,000 base pairs. Then like the minimum thing is from 70. Minimum of 70, you can include your product size and or maximum of 1,000. Okay, and timer turn of 10. That is for general for everything. And temperatures. Melting temperatures are like very important criteria for your primer designing. um, while generating your primers, you need to always check for your melting temperature. It should be of minimum of 52 to 58 percentage. Like it should be in that range. Length should be 18 to 22 or 24. Maximum 22 is fine. It's fine. Remember the thing. The length of the primer should be of 18 to 22. More like that. The maximum is 24 and try to reduce it the optimum region is up to 22 only. Okay, so 18 to 22. And your melting temperature should be of 52 to 58 percentage. It should be within that range. It should not go more than that one, and it should not go beyond below that one. Okay, it should maintain in that 57, 52 to 58. And that is optimum they have given. But that one we can ignore, and maximum of 63. So this by increasing the maximumity of this temperatures and all will cause looping of your own primers. That's why it's preferably we can go for we have 52 to 58 in that region. That's more prominent region. And external and internal selection. For right now, we don't need to consider about this external and internal selection because if you only know about the whole gene sequence, only we can go for this one. Or else we just try to create our own thing. Okay, minimum of intron. If you want to splice some regions, you can include that in that one. If you slice in the sense, in between you will get a stop codon. So there also you have to be very careful about that one. I will while seeing the results, I will tell you where that region is stopping and where it is starting, whether we can go for this one or not. Then primal pair specifically checking parameters. This is about this one, whether you want to use a pair, you want to create your own parameters or automatic, and reference sequences you want to give from which tool that you want to choose. I'll go to the reference sequence mRNA and organisms once again. Then we are using I'm going to create for whole sequence, then I will see this one. And query name is optional. These things are optional things. Primers must have at least two total mismatches. These things are not here. Don't worry much about this region. And amplicon size also no problem. And get primers you have to give. So first let's paste some sequence from human or any other species. Okay, let me download some sequence. Wait a minute. If you have any suggestion for your region also, you can tell me. I can do that for that one also. One NCBI sequence we will do the primers. Okay, I'm taking synthesized one. This is synthesized peptide of, for insulin. I will try to go with this one. After pasting your sequence, your nucleotide sequence, here and all, we are not changing any time because of we don't know much about this protein and because it's not we are going to manually, they're going to do automatically. So we will keep that basic parameters as it is. And preference they have been given for this one. Okay, we'll go with that one. And these things they have given already. We'll just give get primers and see what are the results we are getting and how to analyze those results. it's making our taking time to create our primers. Remember these things. Like they are asking for your Viva question, like how many pairs of like nucleotide sequence, like base pair should be there, and the melting temperature, whether you need to design from 5 prime into 3 prime or 3 prime into 5 prime, and those things you just remembered, okay? And one more thing, GC content is there, like the GC is there, no? Because ATGC is our nucleotide pattern, right? So A and T will form how many bonds, and G and C will form how many bonds with each other. In between AT, like what type of
Two
bond
hydrogen.
is there? Two hydrogen bonds. Between GC, it's 3, right?
3. Yes, ma'am.
Yes. So like it has, if we are while generating our sequence now, if we are having G and C more in the sense, then the binding will be more efficient. So that's why the GC content of your whole primer, it should be of 60%, 40 to 60%. It should not below that 40%, not above the 60%. Okay, because we are only giving 18 base pairs. In that itself, 40 to 50% of your sequence should be covered with the GC content. After that, like the rest of the things will be covered with the 80 content, and up to this 40 or like 40 to 60%, it should be covered with GC content to enhance the binding with your like the primary regions. We have to enhance your primary regions to bind into your own nucleotide sequence. That's it. We are going for that one. Here we have got the results. We'll see like what kind of results we are getting. So if you see here from the graph we are seeing, this is from like the starting region, it's from 1 here and it ends at 600 here, right? So here the primer one, if you are seeing primer pair, like the details it is giving, it has a length of 20, like they have designed primer for 20, and melting temperature it is having 59. So we have told like we should have a melting temperature of about 52 to 58. So it's a bit beyond that one. So we need to check all the other primers and see whether which one is suitable for us. And TC content should be 55%. That's fine. Like up to 40 to 60 it should be there, and 55 fine. If it is there means then that's fine. And the same thing, if you see that reverse primer also, the complementary reverse primer also, it is having a melting temperature of around 59, and GC content is also 55. So like having a similar pattern of reverse primer also. But if I want to generate, so this is only will code for the protein which is from 162 to 420. So what about this rest of the part? 600 up to 600, I don't know what's going to happen after this 420. And what about this before part? Like before 160 amino acid, what are the things it's going to code for this region, and what are the things it's going for this region? So if your guide, like anybody is telling you to find out, you want to amplify your whole sequence up to 600. So if we given primer blast and we are giving automatically, it has predicted some 10 primers for us. We don't know which region is like our domain region. If we know that region, if we know this 240 from 240 to 4 in this my like active site region, I only want to amplify this part, and that's well and good. Then you can go for this primer and see the parameters, like whether like everything is satisfying or not. Let's take the parameters for this one. If we see length is 20, but melting temperature is very bit high, like it's of 60. So we need to careful about, like we need to study the primer again, and the sequence they have given, and GC content is well fine, and the product also 190 is also maximum fine. If I know only this region I want, then I can go for this primer. If you don't know about, you just want to do for your whole protein, then you have to design from here, from 1 to 600. So we have to reduce as much as of numbers we can include in that your protein coding sequence. So then I should start from here. We have to check from here from ATGC. I have to check like manually. I have to do in that case. In that case, I have to manually do design this primer. I have to see the sequence first. Like ADGC pattern will be there, right? So in that ADGC pattern, I have to first ATG as like first thing has to code and second thing has to code and check in between any stop codon should be there up to this much. Like that I have to create. So what I will do first, first 18 amino acids, I will give it in that like another tool is there called Omega analyzer. So manually do that thing. I have to give it in there and check the parameters. If this can be a primer in the sense, will it qualify all the possible what is it, conditions of length, temperature, and GC content. If it is qualifying all the three, then I can go for that one. If it is not qualifying, then again I have to check move from the other one. Like I have to leave the first amino acid, like first nucleotide pair, second nucleotide pair, and from third I have to start. If I am starting from third, first I have to translate this sequence and see whether in between any stop codon is there or not. If any stop codon is there, because if we want to do for the whole protein, right? Starting from here, like if I am starting from third amino acid, like sorry, third nucleotide pair, first two I am leaving, from third I am starting. In between here itself I am getting some stop codon in the sense, then that is of no use. So we need to check in between there should not be any stop codons to stop here. If stop codon is here, if I am complementing in my structure, I didn't worry about this stop codon, then I have generated that reverse complement for this region, then this primer is not valid. It won't amplify your gene of interest. Am I clear to you people?
Yes, ma'am.
Yes, ma'am.
Okay, then let's see like what kind of, here they have given the table of like primers. 1, 2, 3 up to 10 pairs they have generated, and we need to check temperature for everything. So the maximum temperature they have given here is like up to 50 to 60. Self complementary is there or not, and it is checking for self complementary there also. If either like this or primer itself can make a join with this one or not, that is called self complementary. Whether it will complement with these two itself, or it will go and bind with our gene of interest and then start amplifying, or else it will in between like as a round circle formed by binding together itself means, then that is also a waste of money and time both. So the self complementary property also we need to check whether it is self complementing with each other or not, and temperature, GC content. Those things we need to see. Okay, this is how you need to create a primer. So you have to be very sure about your region you want to do. Whether you want to go for full thing or you want to go for the in between region, or you are okay with this results. Okay, you need to find, because this much, if you see this primer 4, it is of very less length. It's like primer 4, right? Some 87 amino acid is going to code for this one. Are you okay with this 87, or you want to study your whole protein? You have to think about that one and act accordingly. This is what like primer blast. This is how you have to create your primers for your gene of interest. Okay, I will give you a set of another sequence also to find out this primer blast. You have to find out like how many primers are there in that one, whether everything is like satisfying that conditions of temperature, length, and like GC content, whether it is satisfying, and like you need to study the whole thing, whether it is satisfying 5 prime into 3 prime n, and the reverse complement is matching with this one or not. Those things we have to see manually. Okay, I mean the primer, like you have a clear idea of how to do the primer blast. Doing the thing is very easy, just like to understand, like you're understanding like well enough, then that's fine. Is it clear for you all?
Yes, ma'am.
Okay, Sadhya, like temperature should be how much with the which range? Sadia Shakil. Are you there or not?
Yes, Mama.
Okay. Temperature should be within which range? I couldn't hear you. Vishal, do you know what type of temperature it should be? Vishal. So Vishal is not there in the class. You should mark him as absent, right? Megha, can you answer? Where is Megha? Megha is there or not? Megha is there. Can you answer the question? Temperature should be in which range? So except few people, none of you are listened, right? Only I have heard answers from Subhash, Arnab, and one answer from Satish. From Satish, yeah, Satish. What about the rest of the people? Whether you are hearing me or not?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Sadia, what about the answer? Do you know or not? My voice is breaking. Is it so? What about
No,
other
ma'am.
people?
No, ma'am.
Okay, you just type if you couldn't able to answer properly to mic, or you can just type in a comment box. Then I can see whether you know the answer or not. Chandini, do you know the answer? Yes. Like that's like the above level, the lower level. Okay. Did you guys hear the answer? Like temperature you should maintain in 52 to 58. Sadia, are you clear? Okay. While generating your reverse primer, what do you have to do? Either you have to go with 3' end to 5' end, or you have to complement it. Which way is better? Divya, can you answer?
Yes, ma'am. Ma'am, we do from 5' to 3' and then we complement it.
From 5' to 3' for which region? Forward or reverse?
Ma'am, for both.
Okay.
Then we can complement it.
Okay. Are you all clear about these primers? You know how to write your generate your primers, right? Shall we move on?
Okay, ma'am.
Okay, what about that self-complementary? It should be there or not? Self-complement should be there or not? Madanki, can you answer?
Yes, ma'am. It should not be self-complementary because it has to go and bind to the our gene of interest.
Okay. If you're clear in the sense, then that's fine. Shall we move on to the next one? Okay, let's see the next one. And in that NCBI toolkit, okay, you just save these results and like interpret your. Then, okay, next thing we are gonna see is like answers. Okay, answers we are going to see. Like that's a search engine. Like that answers is a search engine where you can get all the details of your proteins, like any kind of protein or gene. Like if you want to see like this NCBI, okay, let me share that tab. Okay, if you see here, Antra's Help Center is there, NCBI bookshelf, and Antra's molecular sequence database system is there. So we'll go for this. Okay, if you go for this page, if you see, like it is like a search engine, right? Like how Google works. Like this, that Andres also work, but within that NCBI. So NCBI is holding a bunch of databases and bunch of collections it is having, right? So I want to see like if my protein, like how many data are existing for my protein. So those details we can find out together as a whole. We can see it here. For example, if in NCP they have opened a new column for COVID informations. In the COVID, if you go into this one and see, like the whole data information will be there. Like if you want to see the data information, it is there, and prevention and cure is there, and research information. So this is specially made for this like COVID, right? So in this only we can we can get all the information regarding COVID. So likewise, you want to find out for some other proteins or some other genes. For example, we'll go with a simple one, trypsin. Trypsin is there. You all know about the trypsin. We'll see how many things like data which is having for trypsin. Like so, how we will identify? I want to check for all the databases. If I am going for that NCBI web page, NCBI. If I'm going for the central NCBI page, I want to choose this one. Like either I want a nucleotide sequence or PubMed compound, PubChem. These things I want to specifically choose and check for trypsin. But if I go with this onto search, if I will give information about only the name of that protein, the description I am giving, and I am getting all the details from all the databases. From literature, how many data are there? For PubMed Central, how many papers has been published in this one? From PubMed and NLM catalogs, what are the things it is having? And bookshelf, like in bookshelf, what are the things it is having? And gene, if you go about the gene, like how many total number of genes, like trypsin alone, like not only like one organism. Whichever organism is can able to produce trypsin, all the details of the gene content like will be represented in the, like presented in this one. These are the total amount you can see here. Geo data sets and geo profiles in homolog gene, prop set, and proteins. In the protein databases, you can see whether it is having concept domains, identical protein groups, and protein and family models and structures. You can see it here. In genomes also we are having clinical data we are having and public we are having. So this is a very simple exercises to just to collect your data of your own protein from like NCBI. Like how many things are there, how many things we are going to use in that one. Those things we have to do. That is all about this on-trace. Like we can say this is totally having around 36. How many databases are there? Yeah, it is found in 30 database. Okay, it is the trypsin is having a results of around 30 databases in the NCBI on search tool, and available in various things like literatures, genes, and proteins. In protein alone, it is going from 5,46,321 proteins it is having, similar kind of proteins. Those things we can see here, and it is not having any SNPs for DB synthesis, it is not having any values, and DB variants also it is not having anything, any data, it is not there in that one. And pathways, there is no pathways like have been there for this trypsin protein. Okay, this is how you need to search your, like, if you want to collect the whole information about your like protein or gene, which you want to know where it is present, where it is not present, then you can go for this one. Is it clear about authors? ENTREEC.
Yes, ma'am.
Yes. This is all about authors. So now we have covered that NCBI toolkit, like third experiment, and two things only we have seen. One is ORF finder, blast, and this authors. These three things we have covered, and shall we move on to the next one? How many classes totally we are having? Can anybody type in the comment box?
Ma'am, can you just repeat the question once more,
Like
please?
how many classes we are having total? Like for your exam for December or November?
Yes, I have exam on the December last week.
December last week.
Yes, ma'am.
Okay.
Yes, ma'am, around December 20th. So,
Okay, okay. Okay, up to that we have time, enough time. Okay, no problem. Okay, then okay, we'll see about some blast tools today. And after that we'll stop it, then we'll proceed it in the next class, okay? Because you have already known about blast, right? There is no much explanation needed for blast, I guess. Or else then no problem in that one. Blast find, right? Can I take the blast tools?
Yes, ma'am.
okay, ma'am.
Only like blast, you know, right? You need to find out homologous sequences through your blast sequence. You know about homologous sequence, right? You have already studied in your theory classes what is homologous sequence, what is autologous, paralogous. Those things you have already studied, I guess. Okay, now we'll go into that blast page. Okay, this is the blast page. If you see here, we are having this blast is a very basic tool we are using in bioinformatics to find out your homologous sequences from all the organisms or from all the existing databases. Like it is a very integrated tool with it has been integrated with various databases like GenBank, NCBI, like all the three databases like DDBJ, GenBank, and EMBL, as well as the protein database like UniProt, SwissProt. What else is there in protein databases? PDB, PubChem, not PubChem, PubChem, and PDB is there. SwissProt is there, UniProt is there, and some reference genomes are there. Those things are like integrated with this blast. So we are having some four major like blocks for this one. Nucleotide blast you want to do, or protein blast you want to do, or blast X, whether you want to translate your nucleotide sequence into a protein, or whether you want to translate into your protein into a nucleotide sequence. That is what we are gonna see in this one. So for example, if I'm going for nucleotide blast in the sense, I have to give input as a nucleotide blast, nucleotide sequence, and I will get a similar pattern of nucleotide sequence. The same was for protein. I will be finding here I am finding a similar gene sequence with a similar gene pattern which is existing or not, like homologous gene is there or not. Here I will be finding out the homologous protein is there or not with other species. And if I'm here, I'm going to give nucleotide sequence and I will get the result as a protein sequence because it will translate this sequence, this blast, it will translate this sequence into a like it will search as a protein in the entire query database, and like vice versa for this one, we will give input as a protein and output as a nucleotide. That is about the difference between the Steve blast and blast things. Okay, if you hear genome also, you can directly give, like if you have any idea about your like human genome, which genome you wanna do the search, you can give, or else we can just give our own thing. In the standalone things like other things, what are the like this blast is having. Blast we have seen already, and global element. These things we will see in our latest sessions. Like for global element, we are having another tool called in Emboss. You will probably use that one, but here also the same algorithm they are using, the Needleman's algorithm. For local element, they will use with Waterman algorithm. So here it is there, and CD search in the sense concept domains. If you want to search, you can do. These are like standalone platforms for this blast. Then you can go for this one also. Here we are basically focusing on this. In this experiment, we are basically focusing on these four major tools of blast. You have already done several times blast in your life, like I know that. So just I will give. I won't drag your time much. I'll just give you information about what are the things you need to consider about this blast. Then you can proceed according to your way. So first I'm going with the protein blast. If I'm going with the protein blast in the sense, I need to give, like I wanna, I'm like my main idea is to find out a similar kind of protein sequence, whether like similar kind of, which means like homologous sequence, that I can able to find out in the other species or not. That is for my main aim, right? So I have to give here as a accession number or FASTA sequence. Now you have suggested me a sequence, then I will go for that protein and I will give as a input for that one. Okay, who's going to suggest me some protein name? Any protein?
Ma'am, kinase.
Okay, we'll go with the kinase then. Somebody suggested kinase. Okay, here if you see kinase from trypanosoma is there, cal calveta is there, Thrasophila is there, Drosophila is there, and different species we are having. So we'll go for the first one itself. So if you see here also, here they have given like what are the IDs, like Streptococcus is having, Salmonella is having, Klebsiella is having, Staph is having, and all other taxes is like bunch of taxes are there in this one. Like kinase is present in almost all the species. So we'll go with the Trypanosoma gray. Take the FASTA sequence. I have taken this kinase from that, as that guy suggested. I have taken that one, and I'm going to run blast for this one. So if you see here, what are the things I have to do here? Whether you want to align two or more sequences, you can do. But right now, I'm only going for a single sequence. That is for alignment purpose. So I'm going for a single sequence and find out the similar pattern for that one. In job title and all, it is not needed. Okay, this I have given. Okay, program selection blast B. Then I'm giving blast. Now we can aligning two more sequences I have given like in the previous one. That might be that's causing some problem. Wait. Okay, here now I have to be very careful about choosing a database. Wherever I'm going to choose this, like this kinase, wherever I'm going to search. Either I want to search using model organisms or UniProt, or I want to go for protein data bank. So where this protein data bank will be like involved in that one. Do you guys have any idea where we will use protein data bank? Because this protein data bank is a structural database, right? You guys know that, right? Like protein data bank is a structural database. So why we need, in which condition we will use this protein data bank? Any idea you're having?
Yes. In order to search template sequence for the homology modeling.
Yes, that's true. Like where in that case we will go for the PDB. And what about that Uniprot? Except Subhash, can anybody answer? Okay, well, if you don't know also, that's fine, no problem in that one. Then if you want to go for a kind of similar kind of proteins, not worried about their structure in the sense, then we can go for this Uniprot. Whether you want to find out the domain, motif regions, or any family related regions you want to find out, then you can go for Uniprot KP. So for right now, we will just apply for this Uniprot, or you want to go for not fine, but the data will be more. In order to reduce the time consumption and the search, like purify the search, then we will go for a specific database. Then we'll go for this BLASTP. The algorithm we're going to use is BLASTP. There is not much work in this one. You just need to understand because we just copy the sequence, paste there, and take the results and analyze the results. But what is happening behind that one, that you have to know by yourself. Why we are choosing is the necessity to do these things to like do that protein. Those things you need to be clear about those talks. Storing and accessing the system is a very like accessing the tool is very easy because it's a very user friendly tool. The only thing you want to see is you just need to know why we are going for blast, why we are going for this blast, or why we are going for nuclear blast, why we are going for T blast, why we are going for blastics. If you're clear in that region itself, then that's fine. You're just going to find out the similar kind of things in that blast with identity, e value, and everything percentage, query covering the your final accession ID. Those things will be in the result page. So like it will take much it's taking much time to show the results. It's taking longer time than considered. Okay, meanwhile we will submit for another nucleotide sequence also. Though it will run. It will run. Then we'll see the results on the any nucleotide sequence you want to suggest. Who's willing? I'm getting one nucleotide sequence. So I'm going to do for that sequence because you guys didn't suggest anything, so I will go with my thing. The pressure one sequence here, and I am choosing the clinical validation database. And organism ID if I want, I can give here. I will exclude that thing. So that you want a highly similar one or not, or more similar one, or somewhat similar sequences. So since you need to choose by yourself, I will go with the somewhat similar sequences that which means like last time. Mm, Include collection. If I see like where from which tool I want to check out this nucleotide sequence. Wanna go for which one? That's up to my wish. Reference sequence I can go. Reference RNA sequence I can go. The sequence I have chosen is also a kind of RNA sequence, so I will go with the reference RNA sequences. Standard database, all the standard database, then submit. Okay, we have got the results for this protein blast. We have got the results for this protein blast. We'll see the results of like what kind of results we are getting in this protein blast. So we have submitted a sequence from a kinase that is trypanosoma grayi. So we are checking out similar kind of like which is of around 20 to 79 amino acid length. Now we are checking a similar kind of thing is existing in the other species or not. That's what we are going to check this one. So if you see here, the species which are like matching with that one they have given right here, like mitogen activated protein kinase, which is from Arabidopsis, Aspergillus, Nicotiana, and again some Arabidopsis, Aspergillus, and Lycosterium. Some different species are there. So in that one, which one is having maximum matches with this one? That is Arabidopsis thaliana. So this is from Trypanosoma. The one which is matching highly with this one is like Arabidopsis. That is to only 98% query coverage, and E value is not like up to that 0 level, and percentage of identity is 36.79%. So which means from 100%, it is only having the identity of 36%. So identity of your sequence is also a bit important in some cases. So if you are generating your templates and all, like for example, if you are building a homology modeling or that sort of things, if you are going to do that thing, your sequence identity should be maximum of 30% and query coverage should be maximum of 60%. In that case only, that template you are using that will be valid as like valid to your work. Or else while creating your own like model, it will form loops all over the protein. So loop second just couldn't able to form second just like alpha, helices, or beta sheets. In those conditions, you need to go for you need to anyway like every time you have to thoroughly check for your percentage of identity as well as your query coverage. Those things you need to be very careful about your work while doing your work. And Akrishna, this is from non-redundant sequences or which one I have given. I have given Uniprot, right? Uniprot. So this all the details are from Uniprot. So I'm given non-redundant. There will be a much more data will be there. So you see here the similarity and the reduced. The Homo sapiens and Saccharomyces, whether we are having 31%, like that we need to check and give. This is how you will do your in alignment also you can see the columns. You can whichever columns you want you can check it up and remove those the other columns and select the sequence. And if you want to do the multiple sequence also you can do for MSA viewer and distance tree results. Graphics you want to do, then whichever format you want you can correlate with this one. So I'm clicking the first one. So it will here it will show you with my study subject. Our sequence and the sequence which we are getting the results. Those things will be aligned here, and here you can see how many gaps they have been inserted here and how many mismatches are there, and a long gap is there. So looking the gaps and the gap opening and gap insertion. Those things are called as penalties, right? Have you heard about this penalty words? You might have studied in your sequence analysis paper. What is this?
Be louder.
Okay, because I'm in lab, that's why I was talking a bit slow. Now am I audible?
Yes, ma'am.
Okay, have you heard of this gaps in your theory paper? Gap insertion.
Yes, ma'am.
extension, those things you have heard, right?
Yes, ma'am.
That's what here they have like mentioning, like we are seeing those things, like they are having a gap, like a gap opening we are having. For gap opening we are having a different penalty, extension we are having a different penalty. Those things you know, right? What are
Yes,
about
ma'am.
those penalties and all?
Yes, ma'am.
These penalties only we are able to calculate their identities in gaps. Like if you see here, so they have given you gaps, like how many gaps are there, and positivities, like what are the things are there, and identities are there, and expected e value. These things you can see here. One by one you can analyze with your sequence. That's all about this protein blast. The same way for this n blast, like that is blast n. This is the another result page of that blast n. Sorry, nucleotide blast, not blast n, nucleotide blast. Here we are checking. We have given an chronin as a nucleotide sequence as an input in this one. So I'm having a covalent length of T166. Molecule type they have given, that is DNA. And the ID I have given is went for which one I have went. Reference sequence. Reference sequence I have went. So that's why the ID, like it is finding out. I have given it is from Mycena. Where what are the other homolog sequence I'm getting is from Serpula, Sulius, Heterobadician, Physiophylum, and Mycena we are already having. Agaricus, Fibroporia, and Sparasis. Those are the different other organisms which have been like those things, or like having a similar homologous sequence with your nucleotide sequence. The same results you can see, but the thing that is protein, this is nucleotide. And here you can see the E value for the first one. If you see the first one, it is the exact same sequence it is showing. That's why it is showing 100% of query coverage and E value with the expectancy of 0% and person's identity. Which means the first sequence is the same sequence that we have given, right? That is the first sequence it has taken, considered as a reference sequence. And for the second, if you see like the variation, like the identity is like decreasing, and we can see the variations throughout the page. Is it clear about and like nuclear blast and this protein blast?
Ma'am.
Is it clear about those two things? Like, are you all clear?
Yes, ma'am.
Okay.
Ma'am.
Yes. No.
Ma'am, for two sequence, in order to show that these sequences are similar, what is the minimum threshold for identity? Like, is it 60% or something?
Okay, that is like you know about the twilight region. If it is going beyond that one, like identity is beyond like below 30 or something, it is having a less similarity, like less homologous similar sequence. More than that means then that's fine. Like it is homologous sequence. If it is crossing the 30 percentage, if it is above the 30 percentage, it is a homologous sequence, no doubt about that. But the identity is very, like that is very less. Like it is homologous, but it is crossing beyond that, like below that one, which means like 27% is there, right? That is up to that maximum. We will say that is a twilight zone or something. We will call it as a twilight zone, which means like below that region, below the 25 or like 20, we can say it's very less homologous, which means like the evolutionary distance between those two sequences is the distance will be very more. Like if we are having that graph or something, let me see whether it can possible to get a graph
Ma'am,
distance
in the
from
previous result, like the identity percentage were, yeah, in
like
the previous example that you are showing, the previous search.
which one
The previous search that you showed, the last one,
like,
kinase, I think that was.
okay, kinase. Okay, let me show that page. Here you want to okay, here.
Yes, ma'am.
Okay, here.
Yeah.
Okay, here if you see these are 36%, which means it's homologous, and query coverage we are having 98%. That is also fine. Below that 20, below that 27, we are saying, right? If we are having that much of like variation only, which we will say that as a less homologous sequence.
Okay, ma'am. Got it.
Our main aim in the blast is to search the homologous sequence, right? So that's what we are doing here. We are checking whether we are having homologous thing we are having with our neighboring species. Okay, I will show you one more page. So if you see here, or all of you are able to see this page, like how will I get this page in the sense, like in this page it is there, no? If I go for this distance tree of results, GenBank graphics distance tree of results, MSA Viewer is there, right? If I select this distance tree of results, then it will show you this kind of page where you can see the distance between your original sequence. This is the original sequence you have given. Partial mRNA, mycena, indicator, coronine I have given. And the distance, the farthest distance from this sequence is this one. Sparacesis coron. This is a different species called Sparacesis crispa, coronal-like protein. It is also a partial mRNA. Now can you able to see the distance between these two things? Like very closely related to this one is the same species like partial mRNA we are having from mycena, and other thing is mycena indigotica with a different ID we are having. Some it is from 400, it is 900. So if you see, this is how we will see the homologous difference between those sequences. Can you able to you guys able to understand why we are doing this blast? Is it clear, Arnab?
Yes, ma'am. Got it, ma'am. Yeah.
Okay.
Ma'am, is it like the sequence that is shown in highlighted part?
Yes.
Showing that with they are in very according to that distance, we'll understand how much evolutionary
Yes.
distance. Yeah.
Yes. Like the highlighted one is our input sequence.
Yes, ma'am.
Okay, and which is very closer to this one is like mycena
I see.
indigotica coronine with a different ID, like which is from same species, the different IDs it is showing. Maybe from and the farthest distance is this one, Sparasis crispa, and the next farthest is this Geophyllum trabeum. Those things. Okay, this is how we will differentiate that thing.
Okay, ma'am.
Okay. Anybody else is having similar kind of doubt? No, in the sense, it's fine. This is now we have seen for blast that the result I have shown you, like that tree result. The tree result is for in nucleotide sequence. Now I will show you that same kind of results for your protein sequence, that is kinase. We have given kinase, right? Now we can see here also a different kind set of a tree it has developed. So if you see here, our trypanosoma gray is in this region, which is closely related to this region what it is showing, like homologous sequence. And this is how this graph has been generated. And the more farthest one is the first one, like calcium calmodin dependent protein kinase type 1, short CAMK from Saccharomyces form line 72H. This is the farthest thing that we have found from this kinase, from this trypanosoma grayi. This one is the farthest one, and this one is the nearest one. These things are the nearest ones. Okay, and here they have given you some colors, color legends. Those things from which organisms, like those things they have given. Primates or frogs, toads, cellular slime models. This blue represents the cellular slime models. This green represents eudicots, other species. And here they have given primates, rodents. Those things are there. If it is highlighting that one, then those are belong to that kingdom or something. Okay, this is for protein. Is it clear for these two things, like NCBI blast and NCBI nucleotide blast and protein blast?
Yes, ma'am.
Yes, ma'am.
Okay, two more are there. Should I teach you or you wanna try that one? by yourself, which is convenient for you.
Which one, ma'am?
Like T Blast and Blast X. Two more are there na in Blast.
Yes, ma'am. We will try.
Okay, let me show you that page possible. If you are trying, then that's fine. If you want me to try also, then I will also try. Okay, this T Blast X and T Blast N is there, right? These two things. Will you try or should I teach you?
Yes, ma'am. We'll try.
You will try now? Okay.
Yes, ma'am.
I will give you the details for those things, like what are the things you have to do for the experiments. I will share you the tab with you. Wait a minute.
Excuse me, ma'am.
Yes?
Ma'am, so NCBI toolkit, that 3 topics is exercise 3,
Yes.
and last this 4 topics is exercise 4, right?
4, yes, yes.
Okay.
Okay, this first two things I have given you already. Okay, these 3. Let me zoom it. You just take a snap of this one, like for exercise number 3, NCBI toolkit. The ORF finder you want to find, like ORF you want to find for this IDs and primer blast for this ID, and address for this protein, dystrophin. Like you just check it out. What are the details? Like dystrophin, if you give like we have done for trypsin, right? So if we are giving for dystrophin, what are the things we are getting? Like how many databases it is having this kind of details. Those things you can see in that one. And blast tools for blast P, you try this one, and blast N for try this one. Blast X and T blast X also you try these IDs. Still, if you couldn't able to understand that thing means, then again I will teach you. Okay?
Okay, ma'am.
Okay, you just take the screenshot of this one, and the first two things you are having right already. These two, emboss and expressy tools. I have
Yes,
changed
ma'am.
some IDs in Divine. I didn't change it here. I have given it to Subhash. If you are couldn't able to find out any ID, then you just tell me, mail me, or anyway, mail only you are having, right? Okay, just mail to my ID, then I will change that ID. Okay, if you are facing any problem in ID problems. Okay.
Okay, ma'am.
Shall
Okay.
we finish the class?
Okay, ma'am.
And the next class, I will see everybody's work. Okay, just be ready with your works. All the other people, those who didn't finished, like those who didn't finished, Nishi and some other
Yes,
people,
ma'am.
Satish or someone, like whoever didn't finish, you try to complete it soon. Okay.
Okay, ma'am.
By next class, I will go through your paper, like through the screen. We'll see. Shall I finish? Shall I end the call? Thank
Yes,
you.
ma'am. Thank you, ma'am.
Thank
Okay,
you,
Thank
ma'am.
you.
ma'am.
Thank you, ma'am.
Thank you, ma'am.
Okay.